Hello, everyone. My name is Darren Loy, and I'm the President and Executive Director of Aptorum Group. Today, it is a pleasure here to present to you a little bit about Aptorum Group as of our Q3 updates for our company. Just before I dive into the presentation, if you were to consider investment in our company or otherwise, I urge you to read the following disclaimer, which contains a number of information, which contains forward-looking statements in our below presentation itself. Aptorum Group is a biotech company. We focus on a number of what we call unmet medical needs, including areas of development in orphan diseases with an oncology focus, as well as infectious disease space, as well as a number of earlier stage discovery projects in the metabolic diseases and women's health area. And as a result of that, we've culminated over 15 therapeutic candidates under active investigation or under active development as a result of both our proprietary discovery platforms as well as third-party collaboration approach with other academic institutions. Our business strategy is to focus on early stage discovery. So this is potentially even looking at candidates that have the potential to become great drugs and then finance and develop these into what we call the phase 2 proof of concept styles. After which we have a number of options available, including to continue to develop the pivotal trials ourselves or to explore other co-development opportunities with other pharmaceutical companies itself. Our regulatory focus is the US FDA. Following the US FDA's validation, we also pursue other major markets, including the China markets, as well as the European markets as well. Our company is listed on the NASDAQ since 2018, and we're traded under the ticker APM. And recently in July, we were also cross-listed on the Euronext, also traded under the ticker APM. Our company's principal office is based here in London, in the UK. However, we do have a number of development sites, including one in Toronto in Canada, which covers our major work stream, including, for example, GLP studies, GMP manufacturing, as well as for some of our clinical trials conducted in North America. It acts as a clinical trial coordination hub for our programs itself. Currently, as a whole, the company, we have about 40 full-time staff. And also, in addition to that, we work with another 45 or so scientists, consultants, and advisors in their respective field of expertise and interest. As I said, my name is Darren Lui, and I'm the President Executive Director. My background has been mostly to do with the capital markets. Before that, I was a graduate from the Imperial College of London, majoring in biochemistry, after which I worked for a British bank called Barclays Capital for well over 10 years, accumulating a total of over 13 years of global capital market experience. Our chief founder and executive officer is Mr. Ian Kuhn, who was a graduate from Princeton University majoring in economics, after which he joined Janus Mutual Funds as a US healthcare equity research analyst, following which he has 15 years of global asset management experience. Our chief medical officer, Dr. Clark Chen, attained his MD training in UCL in London, subsequent to which he attained his MBA qualification in University of Iowa, with over 10 years of working experience in Raffles Medical Group, where he served as a deputy general manager. Our head of research, Dr. Thomas Lee, spent most of his career with Novartis and Celgene. And during his days in Celgene, he was part of the candidate selection committee. And over his portfolio, he was in charge of overseeing 25 assets under development, of which 5 of them has reached the market and a number of which has exceeded $1 billion in sales. We're recently joined by Dr. Herman Weiss, who's the CEO of one of our subsidiary platform called Clavis Life Sciences, focusing on metabolic disease research. He also serves as senior medical advisor of Aptorum Group. Based in Israel, Dr. Herman Weiss brings with him 20 years of experience in the medical field. He was trained in MD in Ohio University, after which he attained his MBA in George Washington University, after which he spent most of his career as head of clinical development and medical affairs at Juniper Pharmaceuticals, before the company was subsequently acquired by a much larger pharmaceutical company. We're recently joined by Dr. Kira Shineman as a senior strategic consultant. Dr. Kira Shineman is the founder of one of our own company, Bacteraemia Biosciences. Currently, she serves as a managing director at Healthcare Investment Banking Group, H.C. Wainwright. Before that, she attained her PhD in Mount Sinai School of Medicine and also her MBA from Sickler School of Business as well. We are joined by Dr. Robbie Marshner, who is the assistant professor of pediatrics in the Stanford University Medical Center. He attained his MD in Harvard Medical School and currently serves as senior scientific advisor to our leading program, SCCT1, targeting neuroblastoma itself. Here, I would like to share with you a quick snapshot and summary of our core programs. Currently, we have, as I said, we have over 15 candidates under active investigation, of which 2 of the assets we're currently prioritizing our resources on in bringing them close to the next clinical trial stage. The first asset is SACT1 program, which is targeting a disease indication called neuroblastoma. This asset originated from our proprietary repurposed drug discovery platform called SACT. Here, we were able to matchmake over 2,600 or so FDA small molecule drugs, and these are approved drugs for a potential in search of a possible drug that could be repurposed against neuroblastoma. Culminating 18 months of work, we identified one candidate which showed efficacy for against this neuroblastoma disease, which we'll talk about in later slides. And as a result of that, we are now in the process of preparing our pre-IND submission, of which we're targeting a Q4 IND submission to the US FDA in order to commence what we call the phase 1b/2a trials in 2021. Under this program, under this series, we also have other programs currently in vitro validation stage, including SACT2, 3, and a program focusing on coronavirus research as well. Our second platform is the Acticus series platform, which is focused on infectious disease research. Here we have our leading asset, which is ALS4. It is a novel chemical entity, a new chemical entity in small molecule form, which we have developed a first-in-class oral formulation to target a very deadly bacteria called Staphylococcus aureus, including MRSA, or some people know that as the superbug. Now, incidentally, MRSA is classified as a high priority development target by the WHO and a serious issue classified by the CDC itself. ALS4 currently is in completion of its IND enabling studies, and we are expecting to be able to submit for IND submission in order to start the phase 1 trials, which will be conducted in Canada starting in January 2021 as well. Currently, under this infectious disease platform, we have 3 other assets under active investigation and development, all of which are in under lead optimization stage, including our ALS1 asset, which is focused on influenza virus A, and ALS2 and 3, which is focused on more broad spectrum gram-positive bacteria group. Now, in particular, I'd like to mention that ALS2, 3, and ALS4 pursues a completely novel approach towards targeting our bacteria, specifically designed to overcome antimicrobial resistance issues in mind. In fact, all 3 assets pursue what we call an antivirulence approach, a completely novel approach towards dealing with these very deadly bacteria, which we can talk a little bit about in later slides. Our earlier stage programs include our Clavis platform, which is a microbiome-focused research and development platform, in particular modulating the chemical signaling in our microbiome to target some of these metabolic diseases. This platform is currently led by our new appointed CEO of Clavis, which is Dr. Herman Weiss. In particular, CS1 is a leading program in lead optimization stage right now, targeting obesity. We expect that sometime over in 2021, this asset will be able to reach IND enabling stage. Some of our other earlier programs includes our women's health program, which is the NLS series, of which there are 2 assets in that platform, one of which is NLS1, targeting a highly unmet women's disease called endometriosis. This is a completely novel chemical entity. It's a small molecule that targets women's endometriosis in a non-hormonal manner. This program has completed lead optimization stage and will be moving on to the next stage of IND enabling studies. Our second women's health asset is a supplement called NLS2. Here in particular, the supplement is to target a woman undergoing menopause, as well as a side effect, which is usually associated to menopause, which is osteoporosis. This particular supplement is currently under registration in Asia, UK, and Europe in 2020, and currently commercial manufacturing has already commenced in Canada, and we expect to be able to commence distribution towards the end of Q4 of 2020 as well. I'd like to walk you through briefly our platform of SMART-ACT drug discovery platform. As I said, here we focus on what we call orphan diseases, rare diseases, and currently there are over 7,000 orphan diseases identified around the world, of which 90% is currently untreated or does not have effective treatment. When we actually come to select the disease for screening, we usually take into account a number of factors, including market size, disease knowledge, at the unmet needs of this particular disease, as well as the competitive landscape. and after which we deploy what we call a computational-based screening model, which is depicted here on this slide between step 1 and step 4, using a high-throughput in silico-based screening method of over 2,600 FDA-approved small molecule drug library. After we've identified a promising hits, we will move on to step 5, which is typically in vitro validation as well as in vivo validation, before we subject it to a very stringent IP selection process before moving on to the further clinical trial development under the US FDA's 505(b)(2) pathway. In particular, our leading asset is neuroblastoma, which is depicted by our project SACT1. Neuroblastoma is a very predominant in children, which is a solid tumor, which is related to a nerve tissue cancer. Currently, the 5-year survival rate is around 40%, and with an estimated market size in 2017 of $2.6 billion and a compounded annual growth rate of 3.7%. These are some of our earlier stage results, which we have announced in the market during our in vitro screening. We were able to demonstrate here on the right-hand side that SACT1 was visibly able to show efficacy against neuroblastoma cells in our in vitro analysis. Again, here, this is data we released earlier in the year regarding our in vitro studies when SACT1 is combined with some of the standard of care chemotherapy. And we were here able to observe a synergistic impact when SACT1 is combined in use with standard of care chemotherapy, which is depicted by these yellow and green numbers here on the right-hand side. One of our recent announcements a couple of weeks ago is our latest in vivo study models, including a SACT1 in combination with standard of care chemotherapy versus our control group, which is standard of care chemotherapy alone, depicted here by the blue line. Here you'll see the green line where SACT1 is being applied was able to show after a 22-day study a more than 54% reduction in tumor size. In addition to that, in earlier time points between day 0 and day 7, we were able to observe that SACT1 is able to have an accelerated impact here depicted by this downward sloping green line of early efficacy effect of the standard of care chemotherapy when in use with SACT1 itself compared to the upward sloping blue line. In other words, this is a very promising in vivo studies and supports our continued development into the next clinical stage. As part of our announcement a couple of weeks ago, we also started screening SACT1 on an in vitro basis against 300 other cancer cell lines where we were able to observe a similar efficacy in our in vitro studies. Currently, our company is actively investigating and moving on to the next stage of in vivo studies for other cancer cell types, including but not limited to colorectal cancer, multiple myeloma, lymphoma cancers in the presence of SACT1. As I said, SACT1 is a repurposed drug which has previously established extremely good safety and tox record, including a very high dose of 100 milligrams per day, resulting in 0% death and a fairly long half-life of 43 to 55 hours. Now, I would like to present to you our next leading asset, which is ALS4. Now, ALS4 targets a deadly bacteria called Staphylococcus aureus, including MRSA. We apply what we call an antivirulence approach, targeting this, in particular, a number of MRSA indications, including but not limited to MRSA and bacteremia, which currently we're seeing a 30% average mortality rate and a global market estimated in 2016 of over $2.9 billion with a compounded annual growth rate of 3.2%. ALS4 is targeting a particular defense mechanism of MRSA itself. MRSA is protected by a golden pigment here depicted by Staphyloxathin, which has the effect of protecting MRSA from our neutrophils' ROS attack. And this is the defense mechanism that's what makes MRSA so resistant to our many drugs and also as well as our own immune systems cleansing itself. ALS4 actually targets one of the enzymes that is responsible for the production of Staphyloxathin. And in the presence of ALS4, you'll see here in step 2, MRSA actually loses the protection of Staphyloxathin, and as a result of that, it becomes particularly susceptible to our own host immune system's cleansing through its reactive oxygen species attack. As a result of that, on the right-hand side, you'll see here visibly that as you increase the dose of ALS4, the golden pigment is starting to reduce into what we call a translucent sample here on the right-hand side. In some of our earlier stage MRSA bacteremia study here, we were administered the mice with a lethal dose of MRSA. And by day 4, the black line control group shows that 100% of mice death in our in vivo model. And through the additional ALS, a statistically significant basis, over half the mice survived. Again, in some of our earlier non-lethal bacteremia model, after a 14-day of infection, ALS4 was applied to our mice group, resulted in 99.5% reduction in bacterial count. Some of our results that we announced recently, including a head-to-head comparison to vancomycin in our in vivo studies, where we were able to show that ALS4, which is depicted by compound IM032 here, depicted a more superior reduction in bacterial count across major organ types compared to our first-line defense, which is vancomycin. And results we announced a couple of weeks ago, including our second indication, which is MRSA skin wound infection model in our in vivo studies. Here, we were able to demonstrate our ALS4 compound IM032 showed statistically significant superior skin recovery rates of over 60% recovery relative to the other first line of defense, including linaclid, as well as mupirocin, which has only achieved a low 40% wound closure rate at day 7. And you can see here on the right-hand side, this is pictorially depicted on the right-hand side throughout day 0 and day 7 studies as well. On this basis, I'd like to conclude my presentation to Aptorum Group. Would you like to require more information, please visit our website. It's ir.aptorumgroup.com, or send us an email at investors.relations@aptorumgroup.com as well, here depicted on the bottom of the page. Thank you very much for your time.